Hello friends, welcome to PharmaChem Insights. Today topic is related to ICH guideline Q3A R2, that is impurities in new drug substances. So let's get started. First, what is an impurity? An impurity in a drug substance is defined as any component of the new drug substance that is not the chemical entity defined as the new drug substance. Let us see classification of impurities. Impurities can be classified into following categories. First is organic impurities, that is subdivided into process and drug related. Process impurities arise from the manufacturing process, while drug related impurities develop during the storage. Second is inorganic impurities. Third is residual solvent. What is organic impurities? Organic impurities can arise during the manufacturing process and storage of the new drug substance. They can be identified or unidentified, volatile or non-volatile, and include starting material, byproducts, intermediates, degradation products, reagents, ligands, and catalysts. Inorganic impurities. Inorganic impurities can result from the manufacturing process. They are normally known and identified and include reagent, ligands and catalysts, heavy metal or other residual metals, inorganic salts, other material, example filter aids and charcoal. Residual solvent. Solvents are inorganic or organic liquids used as vehicles for the preparation of solutions or suspensions in the synthesis of a new drug substance. Residual solvents in pharmaceuticals are defined as organic volatile chemicals that are used or produced in the manufacture of drug substances. Let us see rationale for the reporting and control of impurities. For organic impurities, the actual and potential impurities most likely to arise during the synthesis, purification and storage of the new drug substance should be summarized. For inorganic impurities are normally detected and quantified using pharmacopoeial or other appropriate procedures. Carryover of catalyst to the new drug substance should be evaluated during development. For residual solvent, the control of residues of the solvents used in the manufacturing process for the new drug substance should be discussed and presented according to the ICH Q3C guideline for residual solvent. Let us see analytical procedure. Analytical procedures should be validated and demonstrate that it is suitable for the detection and quantification of the impurities. Reference standards used in the analytical procedures for control of impurities should be evaluated and characterized according to their intended uses. Let us see the listing of impurities in specification. The specification for a new drug substance should include a list of impurities. The selection of impurities in the new drug substance specification should be based on the impurities found in batches manufactured by the proposed commercial process. Specified impurities can be identified or unidentified. In summary, the new drug substance specification should include variable applicable the following list of impurities. For organic impurities, following is the list: each specified identified impurity, each specified unidentified impurity, any unspecified impurity with an acceptance criterion of not more than the identification threshold, total impurities, inorganic impurities, and residual solvents. Let us see identification, reporting, and qualification of impurities. Quantitative results should be presented numerically and not in general terms such as complies, meets limit, etc. Below 1.0%, the results should be reported to two decimal places. Example 0.06%, 0.13%. At and above 1.0%, the result should be reported to one decimal place. Example 1.3%. Results should be rounded using conventional rules. Following is the table for maximum daily dose, listing reporting, identification and qualification threshold values. For maximum daily dose, less than 2 gram per day, reporting threshold is 0.05%. For identification threshold, it is 0.10% or 1 mg per day intake, whichever is lower. For qualification threshold, it is 0.15% or 1 mg per day intake, whichever is lower. For maximum daily dose greater than 2 gram per day, reporting threshold is 0.03%, while for identification threshold and qualification threshold, it is 0.05%. Any impurity at a level greater than the identification threshold should be identified. Any impurity at a level greater than the reporting threshold and total impurities observed in these batches of the new drug substance should be reported. All impurities at a level greater than the reporting threshold should be summed and reported as total impurities. All impurities at a level greater than the qualification threshold should be qualified. These are the list of common terms used in this guideline. Let us understand each term. Impurity. Any component of the new drug substance that is not the chemical entity of the new drug substance is called an impurity. Identified impurity. An impurity for which a structural characterization has been achieved is called an identified impurity. Unidentified impurity. An impurity for which a structural characterization has not been achieved and that is defined solely by qualitative analytical properties, example chromatographic retention time, that is RRT. Specified impurity. An impurity that is individually listed and limited with a specific acceptance criterion in the new drug substance specification. A specified impurity can be either identified or unidentified. Unspecified impurity. An impurity that is limited by general acceptance criterion but not individually listed with its own specific acceptance criterion in the new drug substance specification. Potential impurity. An impurity that theoretically can arise during manufacture or storage. It may or may not actually appear in the new drug substance is called potential impurity. Enantiomeric impurity. A compound with the same molecular formula has a drug substance that differs in the spatial arrangement of atoms within the molecule and is a non-superimposable mirror image is called an enantiomeric impurity. Impurity profile. A description of the identified and unidentified impurities present in a new drug substance is called impurity profile. Polymorphic forms. Different crystalline forms of the same drug substance is called polymorphic forms. This can include solvation or hydration products and amorphous forms. Reporting threshold. An impurity above which an impurity should be reported is called reporting threshold. Identification threshold. A limit above which an impurity should be identified is called identification threshold. Qualification threshold. A limit above which an impurity should be qualified, qualification threshold. Thank you. I hope you like this video. Please like, share and subscribe my videos and also show your support in the comment section. Bye.